Molecular docking is a computational technique widely used in drug discovery and structural biology to predict the interaction between two molecules, typically a small molecule, ligand, and a biological macromolecule, usually a protein or nucleic acid. The goal is to predict the optimal binding mode and affinity, which helps researchers identify potential drug candidates or understand biological processes. 1. Introduction to molecular docking. Molecular docking involves simulating how two molecules fit together in three-dimensional space. It predicts 1. Binding orientation, the spatial arrangement of the ligand in the active site of the target. 2. Binding affinity, the strength of the interaction between the ligand and the target. Applications: 1. Drug design and discovery. 2. Protein-protein interaction studies. 3. Enzyme-substrate interaction analysis. 4. Mutational analysis of target proteins. 2. Key concepts in docking. A. Receptor and ligand. 1. Receptor. The biological target, typically a protein, e.g., enzyme, receptor protein. 2. Ligand. A small molecule that interacts with the receptor, e.g., drugs, inhibitors, activators. B. Binding site. The binding site is the specific region on the receptor where the ligand interacts. Identifying it is crucial for docking. C. Energy terms. Docking simulations calculate the binding free energy, delta G, which includes 1. Van der Waals interactions, non-covalent forces between molecules. 2. Electrostatic interactions, attractions, repulsions between charged regions. 3. Hydrogen bonds, bonding between donor and acceptor groups. 4. Solvation effects, influence of water or solvents. 3. Types of molecular docking. A. Rigid docking assumes both the ligand and receptor are rigid. Simplifies computations but may miss flexibility-induced conformations. B. Flexible docking allows flexibility in the ligand and sometimes the receptor. It is computationally intensive but provides more realistic results. C. Blind docking. When the binding site is unknown, the entire receptor surface is scanned. D. Covalent docking simulates the formation of covalent bonds between the ligand and the receptor. 4. Steps in molecular docking. Step 1. Preparation. 1. Receptor preparation. Remove water molecules, add missing atoms, optimize protein structure. 2. Ligand preparation. Generate 3D structures, optimize geometry, and assign charges. Step 2. Defining the binding site. 1. Use experimental data, e.g., crystal structures, or computational tools, e.g., cavity detection algorithms. Step 3. Docking simulation. 1. Place the ligand into the binding site and simulate interactions using scoring algorithms. Step 4. Scoring. Docking tools assign scores based on predicted binding affinity. Common scoring functions include 1. Empirical scoring, based on experimentally determined interactions. 2. Force field-based scoring, uses physics-based potentials. 3. Knowledge-based scoring, derived from statistical data. Step 5. Post-docking analysis. 1. Analyze docking poses and binding energies. 2. Validate through experimental methods like crystallography or mutagenesis studies. 5. Algorithms in molecular docking. Docking programs rely on efficient algorithms to explore possible binding poses. 1. Systematic search, exhaustive search of all degrees of freedom. 2. Stochastic search, random sampling of poses, often used in flexible docking. 3. Molecular dynamics, MD, simulates the receptor-ligand complex's movement to refine poses. 6. Popular molecular docking tools. A. Commercial software. 1. Schrödinger Glide, offers precise and flexible docking with high accuracy. 2. MO, Molecular Operating Environment, integrates docking with molecular modeling. B. Open source tools. 1. Autodock, widely used for flexible docking. 2. Autodock Vina, faster than AutoDock with improved scoring functions. 3. Swiss Dock, web-based docking for beginners. 4. PYRX, integrates docking with visualization tools. 7. Challenges in molecular docking. 1. Protein flexibility. Proteins often undergo conformational changes upon ligand binding. 2. Water molecules. Solvation effects are challenging to model accurately. 3. Scoring function limitations. Existing functions may not always correlate with experimental binding affinities. 4. Computational cost. Large-scale docking or flexible docking can be resource-intensive. 8. Advances in molecular docking. Machine learning integration. AI-driven models predict binding affinities and improve scoring functions. B. Quantum mechanics, molecular mechanics, QM, combines quantum mechanics for the ligand and molecular mechanics for the receptor for greater accuracy. C. Docking with cryo-M. Data docking into high-resolution cryo-M maps enhances understanding of large complexes. 9. Validation of docking results to ensure reliability. 1. Retrospective validation. Test docking methods on known complexes. 2. Experimental validation. Verify docking predictions through in vitro or in vivo experiments. 10. Conclusion. Molecular docking is an indispensable tool in drug discovery, offering insights into molecular interactions and guiding experimental studies. As computational power and algorithms improve, its accuracy and applicability will continue to grow, bridging the gap between computational predictions and real-world biological phenomena.